Okay, so we handled the large-scale, the DNA assembly and delivery session that we had split. And really, there was a lot of complementarity between the two sessions that I hosted and the one that George hosted. Along the continuum from assembly to delivery, we focused a lot on the assembly, both the pinch points and the opportunities. And George really focused a lot on the downstream assembly, obviously with both of us looking at the applications, implications, if you will. And really, I think one thing that galvanized a lot about our portion and our sessions was Jim's comment this morning about synthetic biology as a tool to address interesting biological questions, and we certainly heard a lot of that in the session today. So let's start with the vision. In 5 to 10 years, we're successful if we're able to ... I think large, very large constructs, complicated constructs, libraries, things with very large repeat units, delivered accurately and certainly in a very short time frame. One day was what was thrown out there as sort of the big audacious goal. And then on the delivery side, it was really being able to transfect the DNA into any cell line with very high efficiency and target to specific genomic sites. And then again, the deliverable may actually be anything from a construct, even a linear construct itself, plasmid, or a cell line itself. Okay, moving to smaller font size here on both the technology push and the application side. Really, on the technology side, really one that certainly came out of our session and I think is not uncommon to everybody in this room, is very much facilitating the drive for people to use versus build. And what I want to be clear here is to separate it from those whose mission it is to develop new methodologies and platforms for DNA assembly and delivery, whether that's conventional fluidics and automation versus all the way to microfluidics, but really to address the vast majority of people that I will call end users. And this includes certainly the base requirements that everybody has, cost, and we've heard this throughout the talks, cost, turnaround time, and again, long and complex constructs, plasmids, you know, the entire continuum of product lines, if you will. And then the other one is really what we've heard a lot about, that people are either developing the rules for or developing by no choice of their own, are things like parts libraries and design rules to really enable the rapid turn of pathways and circuits that people are working on downstream. And then certainly as these technologies would push forward, what we have to make sure that came out of my session was really keep our eye on both existing and new screening technologies to match the synthesis scales that, that we would be able to provide, both in terms of diversity of the screening technologies as well as just the throughput if people are able to now work with hundreds of constructs, whereas previously they've only been able to get singles or tens. And then lastly was really supporting the diversity of application drivers, and we've heard these certainly repeated quite a bit throughout yesterday and today. Again, we've heard about DNA, RNA, and protein as all of what people are working with. A lot about in vivo, and today also we heard a lot about in vitro. Again, discovery through diagnostics, whether it's a diagnostic test, discovery of therapeutics, and certainly delivery vehicles is what we heard. We heard about vesicles today, but we can't lose sight of that. And then again, transfection methodologies for variant cell types, whether they be yeast, E. coli, or the mammalian that has been the focus of yesterday and today. On the application pull side for what we need, first, we have the resurrection of extinct species. I'll let everybody's mind wander as to whose session that one came out of. So we've got that one, and pick your species, if you will. Enter the Neanderthal. Okay, certainly changing the genetic code. So instead of responding, how do we make humans resistant to viruses? Humanized cell lines that grow faster for reproducing therapeutics. Again, that taps into a lot about the in vitro versus in vivo. Large genetic constructs with variations, and then certainly delivery for therapeutics. And then if we look at what the main challenges and roadblocks in assembly and delivery are, again, we hear the repeated themes, speed and reliability in synthesizing or receiving if you're the end user, complicated sequences. Hairpins, AT, GC-rich, repeats, certainly a lot of that where we heard a lot of great applications of zinc fingers and talons through the last couple of days. Again, libraries, large libraries with both fixed and variable sequences, and that sort of gets into the parts piece, and that came out in my session. If there are conserved regions and changing regions, how do you do that as sort of parts as opposed to individual constructs to help everything certainly cost and turnaround time. Custom constructs. One that came up in my session was non-natural phosphoramides. One thing that hasn't been in the talks, but one thing that has certainly been in the science literature as of late, people showing that you can replicate DNA with non-natural bases. And then again, as we said, came out of George's session, was really DNA delivery in a very targeted integration with targeted integration and copy number control. Okay, and with that, I'll take a breath, and I guess I've got a couple of minutes for questions. I'm going to make sure the first talk doesn't push everybody behind.
And comments.
All righty.
Okay, good.
Great. Thank you so much.